So welcome everybody to this Ask a Researcher session hosted by the ASXL Rare Research Endowment Foundation. This is a program that's intended for families to help families learn more about some of the current research that's happening in the ASXL space. So our presentation today will be in mostly in layman's terms, so it's a very complicated subject. We will have the opportunity to take your questions at the end of the session. We are very delighted to have Dr. Rosanna Weksberg and Zain Awamleh, who are here to present their work. They are from the Hospital for Sick Kids in Toronto, and they are also recipients of one of the ARRE Foundation's very first research grants. So it's really exciting that we're going to have the opportunity to hear a little bit about some of the work that has been partially funded by the ARRE Foundation, and that funding has come from families like you. So this is a really special opportunity for all of us to hear what our investment in research is making possible. So I am now going to turn it over to Dr. Awamleh and Dr. Weksberg, who are going to lead our presentation.
Awesome. Thank you very much, Amanda. Do you hear me okay?
Yes.
Perfect. Hello, everyone. Thank you very much for the introduction, and thank you for having us. We really appreciate being here and getting an opportunity to tell you more about the research that we're doing. So like Amanda said, we've really tried to make the language as accessible as possible, but even for us as clinicians and researchers, this topic of research is complex. So we really encourage you to ask questions at the end of the session, and even beyond the session, to feel free to reach out to us if you ever have any questions. So I thought a good place to maybe start this talk is to work up to a definition of the word epigenetics, or what it means. So if we think about our genetic code, you might think of this double helix structure. What you're seeing here is the molecular schematic of what our genetic code looks like. It's made out of 4 letters that you see indicated on the screen. They're essentially nitrogen bases that bind to one another in a very specific and complementary manner, forming what we call base pairs. And that's really the basis of this double helix structure that we call our DNA. Now, this DNA sequence is really the code to our growth, our development, and what makes each and every one of us unique. This code is 3.2 billion base pairs long. So there's 3.2 billion of these bases that are paired up together. And this code contains a message that is translated when required at a specific time in our development, or even a specific place in our body, a tissue, an organ. And it's the translation of that message within our sequence that we refer to as the central dogma of life. So it's important to note that changes in our genetic code are common and that they're not always problematic. Some changes to our sequence code are tolerated and really don't have impact on our health. They don't have impact on the ability of the message within our code to be translated. But there are other sequence changes that are more detrimental and can result in disease. And they're often ones that can impact the translation of that message within our code. Examples of these variations are some of the sequence changes that we see in genes causing neurodevelopmental disorders. So sequence changes in the ASXL1 gene that result in boring op-eds. Now, as clinicians and scientists, we have a lot of tools at hand that can help us predict the impact of a sequence change, but they're not always accurate. And with rapid advancement in technology, more and more sequence changes have been identified in more and more genes, but the interpretation of the impact of those changes is not fully, we're not fully capable of doing that. And so much so that there is a group of sequence changes or variations that we've termed variants of uncertain significance. And I will come back to this term shortly. They are sequence changes that we don't understand the impact of them or how they impact the translation of the message in our code. Now, it's important to note that these changes can be sequence changes in our DNA, can be inherited from our parents, and in other cases, they are not. So if we do have a case where there is a sequence change that we observe in an individual that's not present in their parent, we call that sequence change a de novo sequence change, indicating that it's new and was not present in the previous generation. So I told you about our code, so our genetics. Where does epigenetics come in? Well, I told you that this code is 3.2 billion base pairs long. So how is it made to be packaged to fit inside every single cell within our body? And that's where epigenetics comes in. So I'll say epi is Latin for the word above, so above the sequence code. So epigenetics is really referring to the mechanisms that compact our DNA and condense it in order for it to fit into our cells. Here, what I'm showing you is a schematic of how our DNA is packaged. So if you look at the top part of the figure, you can see the letters or the sequence code that I was telling you about, the C's and the G's. And what the DNA does to reduce its surface area is, if you can all see my elastic band here, it kind of hypercoils onto itself. So by coiling onto itself, it's reducing its surface area. And you can see my elastic band folding onto itself. So that's the first way that DNA packages down. The second way is by wrapping around these globes that you see on the figure, the dark blue globes. They're proteins that we call histones. And so the DNA wraps around these histones to further reduce its surface area and compact down. Now, epigenetics is really the mechanisms that regulate the decompaction, relaxation, and the compaction as well of our DNA. And the way that that's done is through depositing chemical marks. So those marks can be deposited in two places. The first place, they can be deposited directly on the DNA, and you can see that indicated in the figure here with the methyl group at the top. I'm going to be talking a lot about this chemical mark or tag for the majority of the presentation. The other place where chemical marks can be deposited is, as you see, on the exposed tails of those histone proteins. And so chemical marks deposited on those histone proteins can trigger the histones to unravel the DNA or condense the DNA. But it's really to come to a conclusion of which message is translated or which code is translated. It's the collection of all those chemical marks that we refer to as our epigenome, whether it be chemical marks deposited on our DNA or on our histones. And this is another schematic that shows you, again, a histone with DNA wrapped around it. It shows you examples of modifications to the histone itself, to the histone tails, and the modification that I'm going to talk about, which is the DNA methylation. And really what I want you to take away from this slide is that those messages are sent parallel messages, and they communicate with one another to come to a final output of whether a message in that region is translated or not. So let's talk about our favorite epigenetic tag that we look at in our research, and it is DNA methylation. So it's the addition of a methyl group directly to our DNA sequence. A methyl group is a carbon and 3 hydrogens, and we know for a fact that it's deposited at very specific sites throughout our genome or our sequence code. And so we're able to map out the sites where this chemical mark can be deposited. To our advantage, this chemical mark is highly stable in cells and tissues, and that's to our advantage because we're able to quantify it. More so, this chemical mark is important because we've identified a relationship between where this mark is located and whether the message that it's located at is really translated or not, whether the code is translated into a message in that location or not. The way we measure DNA methylation is through extracting DNA from blood or tissues and hybridizing them into this microarray chip. This chip really maps out the location of over 800,000 sites in our genome where methylation can take place. And by mapping them out, we are able to compare individuals with neurodevelopmental disorders to individuals that are typically developing, and compare at each site what the differences are in methylation. And so a large proportion of the sites are similarly methylated, but we are really interested in those sites where there are big differences in methylation between the two groups. When we identify a pattern that is specific and where the differences in methylation are so large, we start to refer to it as a DNA methylation signature. A signature is essentially a robust pattern that we're seeing in differences between these two groups. And we say, okay, this is what we want to look at, where methylation is the most different. So in our research in our group, what we have been successful at doing, our group was the first to identify that there's specific DNA methylation patterns that are associated with detrimental sequence changes in specific genes. So for example, one of the first patterns we identified were in individuals that had sequence changes in the gene NSD1. This gene is the gene underlying Soto's syndrome. Another example is sequence changes in KMT, the gene KMT2D, also were associated with a very specific DNA methylation pattern specific to Kabuki syndrome and different from the one that we identified for Soto's syndrome. And really in the last little while, like I had told you earlier, with rapid advancements in technology, there's now sequence changes in more than 70 genes causing neurodevelopmental disorders that have been identified. And interpreting these sequence changes or what they mean can be challenging for clinicians and scientists and can be challenging to provide a diagnosis. And so we've been looking at DNA methylation signatures to use them as a tool to help us interpret some of these sequence changes and what they might mean. Before I go on to my next slide, I want to leave you with this also very interesting piece of information that we identified through our research. Many of the genes that harbor sequence changes causing neurodevelopmental disorders like NSD1 and like KMT2D, we found that they themselves carry a message that is important to maintain the chemical marks or maintain our epigenome. So they themselves function in epigenetics. And with this knowledge that we have of DNA methylation and the evidence, the early evidence we've had of the patterns that we identified in neurodevelopmental disorders, and gearing that tool towards diagnosis, we start to ask the question, how can we apply this to ASXL-related neurodevelopmental disorders? Especially with recent evidence also showing that ASXL proteins or ASXL genes that are carrying the message, they themselves also function in epigenetics. And so based on our previous knowledge, this really encouraged us to start looking at ASXL genes and their DNA methylation patterns. So we measured or profiled DNA methylation in blood using the microarray chip that I had already introduced to you. And we were able to quantify DNA methylation in 14 individuals that had a confirmed boring opids diagnosis, and we know have detrimental sequence changes in the gene ASXL1. And those 14 cases are what helped us establish the DNA methylation pattern that is specific to boring opids. And as I mentioned before, we compare, to identify that pattern, we compare to typically developing controls. At the bottom here, what I'm showing you is what we use as clinicians and researchers to identify the location of the sequence changes across a gene. So this is a schematic of a gene, and it shows each one of these lollipops shows you a single sequence change for a single individual. Oops, it flipped too fast on me. My apologies for that. So back to our gene map. Each one of these lollipops represents an individual and represents a sequence change. What we were able to do is, with using the DNA methylation data for these 14 individuals and typically developing controls, we identified 763 sites where the DNA methylation levels are very different. What you're looking at here is a way to help us visualize those differences. It's a heat map. So I will start by saying that typically developing individuals are indicated in gray, and individuals with boring-opids syndrome and detrimental sequence changes in the ASXL1 gene are indicated in purple. So each column is an individual, and each row is one of the 763 sites that we identified. And the pattern in color helps us identify the intensity in methylation. So the more yellow, the more methylated, and the more blue, the less methylated. So you see almost the heat map split in half, where half of it is yellow for individuals with boring opids and the bottom half is blue. And what's that really showing is about half of those sites have more methylation compared to typically developing controls, and the other half has lower methylation compared to typically developing controls. So now that we've identified a DNA methylation signature or pattern that's very specific to boring opids, we wanted to take that and compare it to DNA methylation profiles of a number of individuals. First, we wanted to look at individuals that have sequence changes in the genes ASXL2 and ASXL3. So detrimental sequence changes in ASXL2 are the underlying cause for Shashi-Pina syndrome, and sequence changes in ASXL3 are the cause of Bainbridge-Ropers. The other DNA methylation we wanted to compare with were any individuals that have variants of uncertain significance in the ASXL1 gene. And I had touched on this term earlier in my talk. So variants of uncertain significance are really sequence changes in genes that we're not able to interpret what they really mean. And lastly, we wanted to compare to other individuals that have neurodevelopmental disorders. So how do we do this comparison? The most efficient way for us to conduct this comparison is to come up with a predictive model, a model that will predict, based on a DNA methylation profile, if an individual resembles more the boring opids profile or if it resembles more typically developing controls. So the first thing we wanted to do is make sure that this model works. And so what you're looking at here on the y-axis is the probability from 0 to 1. The closer you are to the top of the y-axis, the higher the probability that your DNA methylation profile resembles one of an individual with boring opids. If you're closer to the lower portion of the y-axis, closer to 0, that means there's less probability that your DNA methylation profile resembles boring opids. So to ensure that our model works at first, we fed in 100 DNA methylation profiles for 100 typically developing controls and 6 individuals that we know have a confirmed boring opids diagnosis. And what we see is that our model works in that it gives a very low probability for individuals that are typically developing and a higher probability to individuals with boring opids, which is what we would expect. So now that we know our model works, we can start comparing to these other groups that I had mentioned to you. So the first one are 3 individuals that have the variants of uncertain significance. So they have sequence changes in the ASXL1 gene. They have clinical features. However, their clinical features do not resemble boring opids. And so it was very difficult to provide an answer to the parent whether these individuals really have boring opids or not. And with using our tool, we're really able to confirm that their DNA methylation profile does not resemble boring opids. And that's in line with the fact that they are also missing the clinical hallmarks and features of boring opids. Next, we wanted to predict the DNA methylation profile of individuals with sequence changes in ASXL2 or ASXL3. So let's start with ASXL2. We had a single individual that had sequence changes in ASXL2 and a diagnosis of Shashi-Pina syndrome. And this individual displayed a high probability on our graph. Their DNA methylation profile does in fact resemble individuals with boring opids. This is unlike the 3 individuals with Bainbridge-Ropers syndrome that did not have a resembling profile. Now, what that really all means, and this is a phenomenon that we have encountered previously in our research, is that there's an overlapping DNA methylation signature between ASXL2 and ASXL1, whereas ASXL3 and Bainbridge-Ropers likely has a very unique signature to its own. And then lastly, to really make sure that our model or ability to predict whether a DNA methylation profile is boring opids-like, we added 3 groups of neurodevelopmental disorders, Kabuki, Soto's, and Weaver syndrome. For these disorders, all of the genes that are underlying these disorders also have functions in epigenetics similar to ASXL genes. So we really wanted to make sure that our predictive model is specific to boring opids and not any of the other neurodevelopmental disorders that fall within this category. So in addition to being able to help us make sense of some of the sequence changes that we see, DNA methylation signatures are also really cool because they can also help us understand the biology and some of the disease mechanisms underlying these disorders. And so how are we able to do that? So I had already told you that methylation occurs at specific sites in our sequence code, and we're able to pinpoint these sites. By looking closer at these sites, we're able to see which genes they overlap and take a closer look at these genes and what their functions might be. So previously in our DNA methylation signatures, we really have been able to identify genes that function in the immune system, in metabolism, and even in other epigenetic systems, other epigenetic mechanisms, excuse me. In this specific signature that we identify for boring opids, we identified 13 sites out of the 763 that overlapped one specific gene. This gene is called the HOXA5 gene, which stands for homeobox alpha 5. Now we know that this gene is really important, and the way we know that a gene is important is because usually the sequence of that gene is conserved throughout evolution. It's maintained throughout different species, and that's what we see for the HOX family of genes. Now, this particular gene is directly involved in the development of major organs, especially the brain, and plays a big role in body patterning in early life. So with this knowledge that we have and this information that we were able to find recently with our research, this is where our research is headed now. So we know that Bainbridge ropers and individuals with sequence changes in ASXL3 have a unique signature. And so we are really working now towards moving forward and coming up with a Bainbridge roper specific DNA methylation signature. With the boring opid specific signature being available, we continue to utilize it where we are able to confirm diagnoses for certain sequence changes in ASXL1. And we're able to look at those variants of uncertain significance in the gene. And other things we're also exploring is looking at DNA methylation patterns across different tissue types and not just blood. And so really the last thing I want to leave you off with is if you were to think about DNA methylation signatures and really what are the two functions that they serve, I want to leave you with one. They are a complementary diagnostic clinical tool that we can use to confirm diagnoses. And two, they are also useful in helping us understand the biology of the specific neurodevelopmental disorder that we're looking at. I hope I haven't lost you completely with everything I told you. I'm really looking forward to your questions, but would really like to take a moment to thank you all first for allowing us into our community, into your community, giving us the opportunity to do the research that we do, and really participating in it. We would not be able to do our research or the work that we do without the ARRE and the families within the ARRE. So really thank you very much for being a part of our research. And I want to take a minute just to say thank you to some of our collaborators. Dr. Bianca Russell at UCLA has been instrumental in helping us facilitate samples to do this work. Of course, our team at SickKids that has supported me to do this work, especially Dr. Weksberg. And like I said, we're happy to take your questions. Thank you, everyone.
Great. Thank you so much, Zain. Well, I think everyone's probably looking forward to asking some questions. We're going to stop the screen share here and move to questions at this time. So if you have a question, you can raise your hand and I will call on you. And you're also welcome to put your question in the chat and I can read it on your behalf, and also invite everybody to turn your cameras on so we can have some good question and answer here at this time. So if anyone has a question, please feel free to raise your hand. All right, Laura, go ahead. You should be able to unmute yourself.
Okay,
there we go.
Okay, is it working now?
Yeah, so Laura
Okay.
is the chair of the ARRE Foundation, chair and founder. So fitting that she gets to ask the first question. Go ahead, Laura.
Excellent. Thank you so much. This is a really wonderful presentation, very interesting. So I had a couple questions. One was, we know that there's a significant spectrum in terms of clinical presentation. Do you think that if you had a more significant sample of people, for example, with branch opus, that you would see a wider spread in terms of the variation within the precision model, right? So would there be more of a scatter? And then the second question is, when we think about the evolution of drug targets, do you foresee a future with precision medicine where the drug target would be customized based on an individual's methylation pattern?
Rosanna, can I take it?
Sure, go ahead.
Okay, and you can add to more
Sure.
afterwards if I miss anything. So great question. Thank you very much for that question. So you're absolutely right in that if we did have a larger sample size, we could see more of a spectrum in the DNA methylation values. And that's actually something we're working on right now. And Bianca's been instrumental in that. So we are able to identify individuals that have sequence changes that in the ASXL1 gene, and there is variation in the phenotype. And so this is almost the next step. After you identify that there is a specific signature or pattern, now we can say, okay, let's get into that deeper and see if there are specific methylation patterns for certain individuals within that same gene. And that's something we have observed in some of the other neurodevelopmental disorders that we looked at. So it's not a entirely brand new idea for us. So it's definitely within the realm of possibility. And that's what we're currently pursuing specifically for ASXL1. In regards to your second question, and now that I've completely forgot it, that is terrible of me. Would you mind?
That's okay, sure. So as we think about the evolution of drug targets, right, and precision medicine, do you foresee a future where these treatments potentially are customized based on an individual's methylation pattern?
Right. Yes. Thank you. Perfect. As soon as you said the first two words, I'm like, right. That I remember. So again, another area we're pursuing, and that's why I kind of wanted to leave you with DNA methylation signatures. There can be a useful diagnostic tool, but that's what is making them even more powerful, is we're looking at what genes are really impacted. And it's a different lens of looking at these neurodevelopmental disorders. And from that understanding, we are pursuing that information further to get to the point where we are able to develop drug targets for these disorders. So looking at what is underlying the signature is what's helping us identify potential drug targets. And so that it's almost like the research is really driving both avenues of diagnosis and also looking at targets to identify for these neurodevelopmental disorders. Rosanna, please add if you have anything.
Sure, I'm just going to add to this. So there are a couple of ways in which DNA methylation signatures can be used in looking at treatment. The first, and we've developed this in other neurodevelopmental disorders. So for the first one, which is Soto syndrome, where it's a different gene, but also an epigenetic regulator, we've shown that you can, if you correct the gene mutation and so we prefer to do our work in not blood cells, which don't divide, but in induced pluripotent stem cells or neurons. So we've done that with the Soto's individuals, and it's in our plan actually to do it with the ASXL genes as well. But if you fix the gene mutation, you actually fix all the DNA methylation changes. It reverts. So we've actually demonstrated that it's a clear biomarker downstream of the mutation. So not only can it be used as a sensor for drugs in drug screens, which we anticipate we would do in iPSC cells or pluripotent stem cells. But once drugs are identified, there's the possibility of using DNA methylation as a peripheral biomarker for drug response. And also for therapeutics, the DNA methylation becomes very important in terms of entry into clinical trials, because as you suggested at the beginning, there may be people with different levels of DNA methylation changes. There may also be individuals with mutations or changes that actually are in different parts of the gene that don't actually give you the same pattern. And if you included those in a single clinical trial, you would get very messy data. So actually having the DNA methylation signature as one bar for entry into that clinical trial gives you a way to clean up the biology of the individuals entering the trial. So you have cleaner outputs. So there are a lot of different ways in which the signatures can be used in the pathway from diagnosis to treatment, which we're all very excited about. So
Thank you. And just a reminder, you can put questions in the chat or raise your hand. So here's a question from a family that was recently diagnosed. And this is a question about the registry. I think is a good opportunity to sort of talk about some of the samples that you've been able to use in your research. So this family is asking sort of about what happens next after you get consented and included in the registry. And I can provide a little bit more information to this family offline about sort of how to go about doing that. But if you could just sort of talk about the significance of how you've been able to get some of these samples to do this work, and I think help share with families how important it is to participate in research through something like the registry or the biobank.
Yeah, so we, in order to develop those DNA methylation signatures, we need to reach a certain number of samples in order for us to detect that pattern. So we're not able to easily detect a pattern from 1 or 2 samples. We typically actually require somewhere between 10 and 20 samples to be able to identify the pattern and then validate it. And so for individuals that do have a Boring-Opitz diagnosis and have contributed to the biobank, those samples were instrumental in identifying that specific Boring-Opitz pattern. Without those samples, we're not able to identify a specific and robust pattern that we can use as a clinical tool. And then from there, any other sequence changes within the ASXL1 gene, whether they are variants of uncertain significance or Boring-Opitz diagnosis is possible, we use the pattern to help us confirm that it is in fact a Boring-Opitz case, or if it's a sequence change that's resulting in another clinical feature that we need to look at and address. And so I can't say it enough, really. Those samples are instrumental in us being able to identify that initial pattern, and that's really the starting point that we can start to build on from there. Rosanna,
so
please.
I'm just going to add to that, that additional samples are actually critically important, because there is a lot of variation in our DNA and in our biology. So even within the Boring-Opitz group, there's still a lot of variation. And as we build the number of cases that get tested and get signatures done, we can more accurately identify the critical DNA methylation changes that are important for ongoing diagnosis and future treatment studies, and eliminate ones that are variations. And some of those variations are even due to the age of the individual. So it's important to track how the signature changes over time. So there are many variables that we need to look at to optimize the work that we would like to do.
Excellent. So next question is about, coming from the chat, about some of the timing of some of this work. So this is sort of projected way out into the future, but if you could talk a little bit about the time frame potentially of any trials or what technology would be used, and the questions essentially sort of what new technology can bring us closer to drug treatment in this line of research.
Do you want me to take this? Okay. So I don't want to misrepresent where we are. It's very important that everybody understand that treatment is a long way away. That finding the signature is step one. Generating iPSC cells on a number of individuals takes at least a year to generate the stem cells to move forward, and a lot of money. So we are looking at doing this, and although we're very hopeful that we can go down this road, it's going to take a lot of work, and I think we have to look at where we are 3 to 5 years from now in terms of going down that road.
Julie, I saw you had your hand up. Do you have a question?
She's muted. She can't unmute herself, Amanda, maybe?
Okay.
Julie.
There we are.
Thank you. It wasn't allowing me
Yeah.
to unmute myself. I
I'm
apologize.
sorry, Julie.
That's
And actually,
okay.
Julie, will you start by introducing yourself briefly? Because I think it's important for everyone to know who you are.
Sure. So I'm Julie Lopez, and I'm the research committee lead for ARRE. So we have worked with, I've had the pleasure of being able to work with Dr. Weksberg and Dr. Awamleh. I'm sorry, I don't know how to say your last name. Well, I always call you Zain.
Call me Zain. I like Zain better anyway.
Okay. So before, and they've done some wonderful work for us. I actually was just curious to see if you guys had started to see, and I know you have kind of a smaller sample size at this point, but have you started to see any patterns maybe related to, I know they were looking at one point based on where the mutation is on the gene. Have you happened to see any differences in the methylation based on that or anything else?
Yeah, so great question. Yeah, we were talking about this most recently with Bianca, where we have identified individuals that have sequence changes in ASXL1, and you would think based on the sequence change that they definitely have a boring opitz diagnosis, but that has not been the case. We haven't done the analysis on them yet. We're still working on collecting those samples, and Bianca's been instrumental in that. So that also kind of goes back to the first question that came up of, do we see a spectrum in the DNA methylation? And so the answer is likely yes, but we don't have that data yet. So we're working on collecting the samples. It's really important to have all the clinical features related to these individuals so we can connect it back to the DNA methylation. And so I would think that that process would take us a few more months just to coordinate getting the samples, being able to obtain the data and analyzing it. But really, in terms of ASXL1 and boring opitz, now that we did identify a DNA methylation pattern, this is exactly what we are doing, is looking at sub patterns of DNA methylation and what might they mean. Are they related to severity? Are they related to the location of the sequence change? Are they related to, and trying to tease that apart further. I'm sorry, I didn't have a more exciting answer for you.
Yet.
And we do have a couple of questions about participating in the registry, and we do have Dr. Russell on with us, who runs the registry at UCLA. So I'm going to ask her to come on and just explain to families how to go about the process of enrolling in the registry. And there's specifically some questions about providing samples, Bianca.
Can I just interrupt and say two quick things? Is that okay? First of all, I just wanted to let everybody know that we're very excited about the work, and I know that you are, but I want to tell you that this work has been accepted for presentation at the American Society for Human Genetics later in the fall. So that's a big, pat on the back for this work being very important and wanting to showcase it internationally. So that's one thing. And the other thing is I wanted to take my hat off to Bianca and especially to Julie Lopez. So we have had problems accessing samples with ASXL3 from Simons, and Julie Lopez got involved, and we can report now that we now will have access to those samples. And these are samples that were donated by your group, and they are generating stem cells. And so this will really move us forward much, much more quickly. And I really wanted to acknowledge the fantastic efforts of all of your group.
Thank you. Thank you to Julie, and thank you to Dr. Russell. All right, go ahead, Bianca.
She can't unmute herself again. Amanda, I think you might have to unmute her.
There we go. She should be good now.
Yes. Great. This was fantastic. So exciting and really great to hear. And this is exactly the sort of collaborations and the reason that the registry and the biobank were created was exactly for this. So this is super exciting. I think one thing, if I can add to what Zain was saying about looking, and Julie's question about trying to understand if certain changes in the genetic code are linked to different presentations. This is a question that I get a lot too, is my child going to be more mildly affected, or are they going to be more severely affected? And can you tell me that based on looking at their specific letter change? And so I think it's important to notice that one of the challenges with that is that all of these changes that have the signature all are very similar types of changes. They're in the similar location, and they're what we call frame shift or truncating mutations. So they make the protein shorter. And so that's very difficult to try and make correlations between it because there are a lot of similarities in the types of mutations that we see causing boring-open syndrome. So I know that this is a common question I get from the families and something we hope that the methylation signatures can help us with, but I don't know if I bounce it back to you guys and your thoughts on that, but I think that there's some challenges and we may not be able to differentiate things, and that more if one letter change makes someone more mild than somebody else.
So in some conditions, we are seeing definite shifts in signals for milder cases, and in other conditions, we are not. And I don't think we're at the point where we have enough cases for the ASXL genes to know where we're going to end up. So stay tuned.
Every time I mute myself, then I won't let me back. I should just stay unmuted. Thank you. That's great. That helps. So I think when it comes to how to contribute to the registry and the biobank, and I saw in the chat that someone had emailed us, that's exactly the right first step, is to send us an email, and Amanda can share that email as well. And then we can start the process with you of enrolling in the registry. There's a process where you talk to usually one of my research coordinators by phone, and you go through the consent process. We're very close to getting the consent to be on DocuSign, which will be so much easier than having to send things via the mail or scan and print things. So we're trying to make it as easy as possible. And then we'll work with you on a way to work with whatever you have locally to help get samples sent to us through the mail, whether that's going to a local laboratory, going back to your primary care doctor, your geneticist who can help, or the hospital where you're cared for, and help coordinate getting samples drawn. Most of the time what we'll do is actually send you a kit in the mail that has the tubes you need. So you just need to find somewhere to get that done, and we'll help support you in that process. So it does take time. It's a long process to go through all the consents with us and to the coordination. So be patient with us, but also encourage us, and we encourage you to follow up with us if you're not hearing back promptly, because it does take time to kind of go through this process, but we would love to have more people participate in the work that we're doing. And if you're ever in LA or available and can come to UCLA, we can also always coordinate that for you in Los Angeles.
Thank you. So we are at the end of our time here. We have a couple of unanswered questions that I've copied and can share with Dr. Awamleh and Dr. Weksberg offline, and hope to get answers to those two folks. But just wanted to say thank you very much to both of you for all of your work on this. And this is our first Ask a Researcher session, and I think it went well. So thank you all for participating. If anyone would like to stick around to chat, you're welcome to do so. We'll leave the line open until the top of the hour, but otherwise we'll say thank you and farewell to everyone who needs to get going. So thank you, everybody.
Thank you.